Bismillahirrahmanirrahim. Biology 5090, Chapter 6, Video 1. In Chapter 6 on plant nutrition, we have three components. This is 6.1, 6.2, and 6.3. Now in 6.1, basically it's the main concepts of photosynthesis. In 6.2, it's the leaf structure, and in 6.3, it's mineral nutrition. In this video, I'm only going to be discussing 6.1, and then in the second video, we will discuss 6.2 and 6.3. Now, if you look at the first syllabus point, understand that photosynthesis is the process by which plants make carbohydrates from raw materials using energy from light. State that chlorophyll is a green pigment that is found in chloroplasts. State that chlorophyll transfers light energy into chemical energy for the formation of glucose and other carbohydrates. From glucose, cellulose, from glucose, starch. Then number 4, outline the subsequent use and storage of the carbohydrates made in photosynthesis. So the glucose that we made, what is it going to be stored at? Starch as an energy store, cellulose to build cell walls, glucose used in respiration to provide energy, and sucrose for the transport through the plant. So the 4 different ways by which that carbohydrate will be then stored and used. So the subsequent use and storage. Then state the word equation and balanced chemical equation. Investigate the need for chlorophyll, light, carbon dioxide for photosynthesis using appropriate control. So that means we're going to be discussing now 3 practicals in this. Need for chlorophyll, need for light, and need for carbon dioxide. Then describe and explain the effects of varying light intensity, carbon dioxide concentration, and temperature on the rate of photosynthesis. So describe and explain. This is another 3 practicals. Then investigate the effect of varying light intensity, carbon dioxide concentration, and temperature on the rate of photosynthesis. These 2 are linked. Actually, it's this one which has 3 practicals, and the other one is just the describe and explain all that phenomena. Then identify and explain the limiting factors of photosynthesis in different environmental conditions. So we're going to first discuss these in this video, inshallah. Now coming to the equation of photosynthesis, you can see here I've given this in the word equation. So this is the word equation in which you just have to write carbon dioxide in words and water, and then on the arrow you write sunlight and chlorophyll, and then glucose and oxygen is made. But if I ask you for a chemical equation, because both have been asked for, so in a chemical equation you will say, you'll give a balanced equation, 6CO2 plus 6H2O in the presence of light and chlorophyll will give you C6H12O6 and plus 6O2. So there's a 6 here, there's a 6 here, and there's a 6 here, but there's no 6 here with the carbon because this balances out in the equation. Now, what is a chlorophyll molecule? Now, chlorophyll is inside the chloroplast. So chloroplast is the organelle, and inside it you have the chlorophyll inside the chloroplast. So located in the thylakoid membranes are a part of the chloroplast, and chlorophyll has magnesium in the center. Just like hemoglobin has iron, so chlorophyll has magnesium in the center, and chlorophyll pigments harvest energy, photons, by absorbing certain wavelengths of light. So they're the blue and the red ones, and they're most important. And why plants look green? Because green is wavelength is reflected, not absorbed. So this is one thing which you have to understand about the chlorophyll molecule. Now looking at this equation, we have carbon dioxide. Now where is the carbon dioxide coming in? Carbon dioxide is coming from the air and is entering the leaf. It is not entering the leaf, it is diffusing into the leaf. Now why would it diffuse in? Why are we using the word diffusion? Because this, if it's a bright sunny day, if it is a bright sunny day, then carbon dioxide is going to diffuse in. If it's a cloudy day, probably not, it's not going to diffuse in. So it's a bright sunny day, then carbon dioxide is going to diffuse. And why? Because it's going to be a high concentration in the air. And why would it be in the low concentration in the leaf? Because it is being used up for photosynthesis. So low CO2 inside the leaf, high CO2 outside in the air. So carbon dioxide is going to diffuse in from the air into the leaf. Then water, where is the water going to come from? The water is going to come from the soil. Water is going to enter the roots and then come here. So the water molecule is going to come from the soil. Don't say in the roots. The roots doesn't have water. The water is in the soil. So the water is going to come from the soil, and this carbon dioxide and water is going to combine to form a glucose molecule, and oxygen will diffuse out. So why will oxygen diffuse out? Because it's a bright sunny day and the photosynthesis rate is more than the rate of respiration. Respiration is occurring all the time. All plants are respiring all the time. Human beings are respiring all the time. Respiration is the release of energy. Please understand respiration. We'll do it in a later chapter. But carbon dioxide diffuses in. Water is from the soil, enters the root hair cell, and then is carried up and enters the leaf. Here, the water and the carbon dioxide react, and a glucose molecule is made. Now, another thing which we always write is light and chlorophyll. But what we do not write is enzymes. And I can always think of, Cambridge giving you a question on that, because photosynthesis is an enzyme controlled reaction. Without enzymes, this reaction cannot occur. So a whole lot of enzymes are needed for, as I told you in the previous chapter of enzymes, enzymes will be needed for the process of photosynthesis and for the process of respiration, but will not be needed for the process of transpiration, which we study in another chapter. So carbon dioxide, now the trouble is that we have to understand is what is this glucose molecule going to be used up for? The glucose is basically the first chemical. So what do we say? We say light energy. Light energy is converted to, the glucose molecule is classified as chemical energy. So the glucose is the chemical which stores the energy. Chemical, glucose is the chemical which stores the energy. And that energy, of course, is released when you and me eat bread, and that is digested to glucose and enters our body cells where it is respired to release energy. Now, what we need to understand is that glucose is a monomer. It's just this is one glucose, two, three, four, five. But we join them together, then it becomes a starch molecule, and there are hundreds and hundreds of these as a starch molecule. So the plant converts the glucose, it must have enzymes to do that, to convert it into a storage molecule called starch. So just imagine like a 1 rupee note is a glucose molecule, and a 5,000 rupee note has 5,001 rupees in it. So glucose is the monomer, and this 5,000 rupee note is the starch molecule. So this is how I want you to remember is that one glucose unit, 1 rupee. So each glucose is then together in 5,000, in a 5,000 rupee note, you have 5,001 rupees. So this is just to make you understand the glucose and the starch connection. Now from that basic glucose molecule, you can either make a cellulose molecule, or you can make a starch molecule, or you can make a glycogen molecule. In human beings and in animals, of course, it's going to be converted to glycogen. In plants, it's going to be converted to starch, or it is being going to be converted to cellulose. Now you see how they have been joined together. Now the shape of cellulose molecule is a lot of fibers because it gives it strength. But the starch molecule is this structure. It's a branch structure because it has to be compact, and it's an energy store. So it has to be compact. So it's got these branching edges joined together so it occupies less space, but then is easily hydrolyzed or broken down. Similarly, the glycogen also has to occupy less space. Every liver cell of our body contains glycogen. So the same glucose is then put together in such a way that it forms a compact insoluble molecule, but can easily be then broken up and the glucose can be released. Now, basically, your understanding is that from the glucose molecule, number one, starch can be made, which is an energy store. It is that 5,000 rupee note. Or the glucose can be converted to cellulose, which is going to make the cell walls of the plant cells because new cells have to be added. If the plant has to grow, then new cells have to be added, and every cell needs a cell wall. Just like if I want to add another room to this building, then I need bricks. Similarly, the glucose can stay as glucose because it's going to be respired in the plant cells to release energy for growth, for the plant to grow. Or the glucose is going to be converted to sucrose. And what is sucrose? Sucrose is a disaccharide. So glucose and fructose together combine to form a disaccharide which is called sucrose. But why do we need sucrose? Because in the phloem, the glucose, when it is transported from the leaves to the roots, it has to be transported in some way. It can't be transported in an insoluble way. So it is transported as a sucrose molecule in the phloem, and then it is taken to the root because the roots cannot photosynthesize. So they will need the fuel, or they will need the glucose to respire and to release energy, and so the root system can grow. Now we come to the next syllabus point. It says investigate the need for three things. Number one, chlorophyll. Number two, light. Number three, carbon dioxide for photosynthesis. So you have to give me three practicals to prove that chlorophyll is necessary for photosynthesis, light is necessary for photosynthesis, and carbon dioxide is necessary for photosynthesis. And you also have to show me appropriate controls. And now what are controls? Controls are for a comparison. That okay, if you're saying chlorophyll is necessary, now this has chlorophyll, so photosynthesis has taken place, but this part does not have chlorophyll, so photosynthesis did not take place. But how can we prove that chlorophyll? Like for instance, how would I prove that academically you are becoming better? Well, I would give you a test. I would give you a test today, and then I'd give you a test 2 weeks later, and then I would say, okay, fine. Let's see how much you have improved in biology. So, but here in photosynthesis, we have a problem. If photosynthesis is occurring, glucose is made. That we know for sure. But the glucose is soluble and cannot be kept as glucose, so it will upset the water potential. So water will start entering the leaf or the cell. So it is immediately converted to starch. So then, how can we prove that photosynthesis has taken place? If we can test a leaf for starch, and if the leaf contains starch, then of course we say photosynthesis has taken place. If there is no starch present in the leaf, then photosynthesis has not taken place. In every experiment concerning photosynthesis, we have to de-starch a potted plant. And what we do is we keep that plant in a dark room or in a dark cupboard or away from light, or you keep it in a box, and you have watered the soil. The reason is you don't want it to wilt, and you keep in this for 24 to 48 hours. So why are you doing this? The reason why you're doing this is if there is any starch store, that's going to be converted to glucose. The glucose is going to be respired and energy is going to be released. Just like if I say, okay, well, for 24 hours, you're not going to eat anything, but you're not going to die. We're giving you water, but we're not giving you any food. So whatever your food reserves were, your glycogen in the liver will be all used up. And then, of course, your fat will start to be used up. So if I don't give you food for 24 or 48 hours, people have fasted for more than that even. So you can be kept without food for quite a long time, but you will be asked to drink water because water is actually the necessary thing which will keep you alive. But food is not necessary because you have a lot of reserves in your body. So the starch is converted to glucose, the glucose is respired, energy is released, and then after 48 hours, we will set up the practical on this plant in which we have de-starched a potted plant. Now, how do you test a leaf for starch? There are about few steps which we all must know. Number one, you dip the leaf in boiling water. Why do we do that? We want the enzymes to be denatured so all reactions stop. So boiling water is 100 degrees, so hopefully all the enzymes should be denatured because plants do work best at around 20-25. Then the second step is that you put the leaf in ethanol. Now you see this test tube. This has got ethanol in it and it's got the leaf in it as well. So the leaf is the test tube has ethanol, but outside we have a water bath. And why are we boiling this in a water bath is because ethanol or alcohol is highly inflammable and it can catch fire. So you are sort of heating it through the beaker and through the water bath. First the water gets hot and then the heat will reach the ethanol. And then the leaf will be decolorized. Why? Because ethanol is an organic solvent. And chlorophyll will only dissolve out in ethanol. It won't dissolve in water. So you're actually boiling the leaf in alcohol. And the alcohol is going to, the chlorophyll will dissolve in the alcohol or the ethanol, which is an organic solvent. Number three, then you put the leaf in cold water, you dip it. Why? Because we want to soften the leaf. Why in ethanol the leaf will become dehydrated? It will become very brittle. It just become like Lay's crisps. So it will soften the leaf and then you put it on a tile and you add iodine solution to it And if the color turns black, then starch is present. If it remains brown, no starch is present. So this is the standard procedure to test a leaf for starch. Now, the first experiment is, how do we prove that chlorophyll is necessary for photosynthesis? Now, we cannot remove the chlorophyll from the leaves. Please understand that. So what we do is we take a potted plant with variegated leaves. Now, I've given you three different, very beautiful pictures of them. And there are three different, genetically different plants, but they all have variegated leaves. Variegated leaves means those which have white and green patches or yellow and green patches. So the white area does not have any chlorophyll. So the white area in some leaves, of course, there's more white area, like this white area here does not have any chlorophyll. This white area here has no chlorophyll. So, but the green areas have chlorophyll. So there's chlorophyll present here, there's chlorophyll present here, there's chlorophyll present here. So what we do is we take a potted plant with variegated leaves and then we keep it in the dark, so we de-starch the plant, right? Now you expose it to bright light for 8 to 10 hours. Now, if any photosynthesis is going to take place, it's going to take place because, first of all, when you de-starch the potted plant, all the starch was consumed, all the starch was used up. Now you're exposing it to bright light. I didn't say light, I said bright light, because a little light will not really affect it, because photosynthesis will be equal to the rate of respiration. This is what happens at dusk and dawn, which is called the compensation point. So you expose it to bright light for say 8 to 10 hours, and then you take a leaf, you take one of the leaves, so you remove one of the leaf, maybe this one or maybe this one, and you detach the leaf, and then you do the same experiment on it like we did the test the leaf for starch, the 4 steps of it. But before you detach the leaf, you need to make a drawing or you need to take a picture of it to show where are the green parts and where are the white parts, so that you can remember that. Now, when you've done the test for starch, what do you see? It's very interesting. What you see is that the green parts will turn blue-black because they contain chlorophyll. So they will contain starch. So when you do the iodine, the test, the leaf for starch, you've done the alcohol, boiled in alcohol, and then you put the iodine on it. Then you find out that the green areas will be blue-black because the green areas will have converted light energy to chemical energy and glucose will have been made, and then the glucose will have been converted to starch and stored in the leaf. So the green areas will contain starch and will be blue-black in color here, which you can see the green areas. But the area which will be the yellow areas, they'll be unaffected and they'll be colorless, of course, or they'll be brown, which is the color of iodine. So variegated leaf after the starch test. Now the next experiment is to prove that light is necessary for photosynthesis. Now in this you take a potted plant, you de-starch it, keep it in the dark for 24 to 48 hours so all the starch has been used up. Then you cover a part of the leaf, any leaf, one of the leaf with black paper. And then you expose this whole plant to bright light and then you leave it in the bright light for 8 to 10 hours. And then you detach this leaf and then you test for starch. Now, wherever there was the paper clip, wherever there was this black strip of paper, this area here will not turn black, while this area will turn black. Why? Because when you react with iodine, so this area will contain starch, this area will contain starch. So if starch is present, it will convert to blue-black in the presence of iodine. But this area where there was the black paper, this area will remain brown because there will be no starch present. So this fact proves that because light did not reach this part of the leaf, so this part of the leaf will remain brown. This part of the leaf will remain brown because iodine will not, there's no starch in it. So the iodine, color of iodine is brown, so it will remain brown. But these areas will turn black, which were exposed to light and which photosynthesis took place, so starch was made, and then the starch reacts with the iodine. So you do the same steps for the test, the leaf for starch, the alcohol, and the dipping in water and all that, which we did the 4 steps to prove that a leaf contains starch. The third experiment is to prove that carbon dioxide is necessary for photosynthesis. So we take two potted plants, same species, same age, same size, And we have soda lime and we have them in bell jars. And then what have we done is we place the two plants and then we have a chemical. In this we have water. In this we have potassium hydroxide solution, which is going to absorb the carbon dioxide. And it also passes through, the air is passing through. This air does not have any carbon dioxide. The air inside here does not have any carbon dioxide. This is normal atmospheric air. Now we have done the whole thing. We de-starch the potted plants before, then we place them in these bell jars, then we expose them to bright light. And then after 10 hours, we take one leaf from here and we take one leaf from here. So this is say A, this is say B, and then we test for starch. What we find out is that this one does not contain any starch and this leaf contains starch. So this proves the fact that carbon dioxide is necessary for photosynthesis. So one plant in normal air, one plant in air without carbon dioxide. And of course, what do we mean by control? Control means a comparison. So we placed another chemical here, but it's water here. The chemical here was potassium hydroxide, but we had to place another thing here. So this would be the control or the comparison, because I could say, well, it's the chemical there. So we say, no, okay, now there's no chemical, but the same volume of water in this Petri dish. So same plant, everything must be the same. Controls are there for comparison. Now the next topic is how do we study the rate of photosynthesis? Now I always give you this equation and I write it on the board and then I ask you, how would you measure the rate of photosynthesis? And of course you come up with this, we can measure the volume of oxygen produced. So if the volume of oxygen produced in 1 minute is 2 ml, in 3 minutes how much it is in 4 minutes. So we can study the volume of oxygen produced per unit time. Or we can study the mass of glucose produced, but strangely glucose is converted to starch. So we cannot measure glucose because it's immediately converted to starch. So if we can somehow measure the mass of starch in a leaf, then we can, but that's a very difficult thing, so it's not very possible. Or we can study the, if we say we have 100 molecules of carbon dioxide, and now there are 10 molecules of carbon dioxide left. So that means 90 have been used up for the photosynthesis. But then carbon dioxide is also being produced by respiration. So you've got to remember that also, but that's a little tough for O levels, but in A levels, I will suggest you remember that. So the rate of photosynthesis can be measured in many ways. Number one, how much CO2 is being used up, how much glucose is being made, or how much starch is being made, or what is the volume of oxygen being produced per unit time. So usually we're using this one, and this is the one that I'm going to be talking about now, how we can measure the rate of photosynthesis. And what are the factors that we have to change? It says, how would you study the effect of varying light intensity? You see, you don't write, you just write light, and that is wrong biological English. So it is the varying the light intensity. Number two, varying the CO2 concentration. And number three is varying the temperature. So we have to do three experiments and we have to prove whether the rate of photosynthesis increases or decreases when we change the light intensity. Light intensity, remember? When we change the CO2 concentration, does it change the rate of photosynthesis? When we change the temperature, does it change the rate of photosynthesis, or temperature has no effect? So we've got to study these three different variables and we have to study whether the rate of photosynthesis is affected or not. So we will say, okay, how much oxygen is produced when the light intensity was X? How much was the oxygen produced when the light intensity was Y? If the light intensity is increased or decreased, what happens? Similarly, with carbon dioxide concentration, we have to change the carbon dioxide concentration available to the plant in the lab. And then, of course, we have to change the temperature and then study the rate of photosynthesis. Now, the basic apparatus is to be understood first. You have a light, a bulb which has a dimmer switch so you can increase or decrease the light intensity. Then you have a glass heat shield. Why do we have a glass heat shield? Because you have to remember is that heat is also coming out here. And the heat will affect the temperature of this water. So we don't want any heat coming, we only want the light to be coming. So we don't want the, so we place a heat shield in front of the light source. It's called a glass heat shielded, two sheets of glass with water in it. So it allows light to pass but will not allow the heat rays to pass. And then you have a cut stem, or this is a pond weed or an aquatic plant. And in an aquatic plant, it's, we said, Elodia cut stem, and it is a water plant or an aquatic plant. And we placed it under a funnel, and then we've inverted a test tube over it. You can see here, this is the test tube inverted over it. And then by water displacement, the oxygen is being collected here. And you can have the volume of this oxygen if it is a calibrated test tube. Then you can tell the volume of oxygen which is being produced. Now you can change the light. The dimmer switch can help you change the light intensity. You can make it more or less, and then you can see how much is the volume being produced per unit time. So the volume of oxygen produced per unit time, I don't like you say bubbles per unit time, because bubbles is a variable. They can be large bubbles or small bubbles. So a student says 10 bubbles, it could be equal to 5 large bubbles. So that is why bubbles is not a very scientific way to be talking about. So changing the light intensity, we will study the rate of photosynthesis. Now this is another diagram showing you the basic experimental principle by which the rate of photosynthesis can be measured. The important thing which you have to note is, which we didn't mention in the previous slide, is that there is sodium hydrogen carbonate solution to supply the carbon dioxide. Because if you just use tap water in the lab, and that does not contain any chemical. But if this is a pond weed, pond water will be, of course, containing microorganisms or tadpoles or maybe tiny fish or something which is producing the carbon dioxide. But when you've taken tap water in the lab, well, that is not going to contain any carbon dioxide. It's not going to contain any bacteria or anything else in it. So basically, you've got to add a chemical. So this could be a 1% sodium hydrogen carbonate solution. And if you want to study the effect of changing this concentration, that would be, of course, assessing the changing the carbon dioxide concentration. So in one experiment, you use 1%, in another experiment, you use 2%. But please remember, keep the volume same. Volume must remain the same. So if using 50 ml, but then use 50 ml each time. So first time you take 1% sodium hydrogen carbonate solution, then you take 2%, then you take 3%, and of course you study the experiment. Here, of course, you're measuring the length of an air bubble, which is going to help you decide how much is the rate of photosynthesis. The more the length of the air bubble means more the oxygen, means more the photosynthesis. In the third experiment, you have to study the effect of temperature. Now, in that, you will not change these carbon dioxide concentrations. You will keep that constant, maybe 1% carbon dioxide concentration. But what you'll do is you'll change the temperature. So this is a thermostatic water bath. And so you will change the temperature of the water bath. Maybe you have 5 different temperatures. So you say, okay, let's study it at 20 degrees Celsius, then 30 degrees Celsius, and then 40 degrees Celsius. Please keep the range the same. So you will study the effect of changing the temperature on the rate of photosynthesis. I have to end the video here. Thank you very much. Please subscribe and please press the bell icon so that you can be notified whenever there's a new video. The limiting factor from 6.1 will continue in video 2. And thank you very much.